So Carlos Linnaeus, the father, and look at this particular picture. This is his hand notes. Remember, we're talking about the tree of life. So we have the genus and then splitting into different species. This is the gentleman who actually come up with the idea. Very, very nice, interesting idea. And we'll talk a little bit about history, why it was very important. But the main idea, he says that nomenclature, how to name or how properly name the organism, is the foundation of botany. Now it's basically the foundation of biology. And then classification is made, the names should, of unknowing, should have a single genus and a single name. We already talked about several species like Homo sapiens. Homo is the genus, sapiens is the single species name. So yeah, that's coming actually from him. So two different things, taxonomy versus systematics. Taxonomy is basically responsible for naming as well as the describing. You discover new species and there is a special set of rules how you need to describe that particular new species and how you will name it and where it will fall into the group. Is it a plant? Is it a fungi? Is it an animal? If it's an animal, which class it belongs to, and so on and so forth. And then the systematics. Systematics is talking about the biodiversity. How many living creatures do we have? How many different species we have on this particular planet at this particular time? And then, of course, we're trying to link it up some of the modern as well as the extinct species. Like we link it up the dinosaurs, specifically something which known as a lizard hip dinosaurs, the same group as the T-Rexes as well as velociraptors to modern birds. There is a distinct connection to that particular group of the dinosaurs of the modern birds. Speaking of the modern birds, today morning I was walking with the dog around 5:30, around 6 o'clock in the morning, and the birds would have owls living in the wooded area right there. They scared the bejesus out of me and my dog. They start screaming like a very, very loud, almost human-like scream, and then the flapping of the wings, and they were like, you know, mating, etc. So yeah, it was weird. In the morning, early in the morning, nobody's around, and then there is something screaming coming from the wood area I have behind my house. Yeah, it was. Anyway, Aristotle, ancient Greeks, did a lot of things for the history, for the sciences, for the philosophy. So Aristotle come up with the idea of the classification. He classified pretty much everything. For non-being, then he said that minerals are actually being. Why? Because they grow. Yes, we know they're not really alive now, but at that time we didn't know. And he said that they are minerals. Minerals were considered to be kind of living because they could grow. Then, of course, we had plants and animals. Look, a man was above the animals, but below the demons. What was really interesting. And then, of course, angels. And in history, Aristotle has several gods, so there should be gods. And later on, whenever it was adopted by the classification in Europe, there was a single god. So here's was that particular classification was okay for almost 1500 years. No, you don't really have to. It's just a little bit of history, but the one which you will need to be put in the proper order is the actual classification scheme, which I will go to, well, which we are using nowadays. So this is where it started, something like that. All right. So yes, man was above the animals, but below demons. Now we know that man is technically belong to the animals, the beasts, so to speak. And there is a controversy about all of those particular creatures. Do we have demons? Do we have angels? Do we have God? Yes, no, maybe. And which one? If we have a God, there is how many? There is so many religions. Some of them have multiple gods. Some of them have one God, and so on. So we're not going into that. That's a social aspect. We are stopping pretty much at that particular area. Now look at this: animal and plants. Two things. That was pretty much it. Now we know there is more to it. As a matter of fact, animals later on by the Aristotle has been classified into the air dwellers, the water dwellers, and the terrestrial creatures. Now we broke it down a little bit further. So now Linnaeus, Carolus Linnaeus, published in 1735 a first edition of Systema Naturae. Basically, he says that everything will be broken down into that, and he come up with a very interesting way, a standard nomenclature, a standard way how to name it. At this particular time, 1700s, what exactly happened? Why we had that particular need to start naming the creatures? Why we couldn't just name them as we were using before in Europe, let's say in English or in the Swedish, wolf, dog, cat, etc. Well, remember 1700s, we already discovered pretty much all of the continents. We started seeing the creatures which our language at that time, English or Russian or German or Swedish or any other, didn't have a specific name or common name for it. So we started looking at the tremendous amount of species, and we didn't have the idea how to name them. One of the ideas how to name them was to name kind of describing. A tall tree with the small leaves which are with a brown bark and smooth bark and the red fruits or anything like that. But that become very, very messy. For example, a name for a honeybee, a regular honeybee, or containing 16 words. That was too messy. And this gentleman come up with a general idea which we are using right now. First of all, it's all Latin. No matter which language you are or was born into, so to speak, English, Spanish, German, Russian, whatever it might be, no matter what's your native language, the language of science essentially is Latin. All of the species will be named in Latin. That way, whenever I'll say dog in English, it would be perro in Spanish, it would be sabaka in Russian. So lots of misunderstanding. However, if I'll say sabaka in Russian, like I have no idea what to say. But if I'll say Canis lupus familiaris, no matter where the other scientist coming from, they will understand that I'm talking about the family, well, the regular dogs. So in this particular case, we have that particular tiny little box where we put one species. Now, several species usually will put in a bigger box known as the genus, right? And then we'll talk about the families, the classes, the orders, and so on and so forth. So we'll talk about that right there. And remember, humans are considered to be a primate. So this is the classification. What we have, an example is the dog. A small box, dog, Canis lupus familiaris, will belong to the species. It's a subspecies. Dog is actually a subspecies of the wolves. So in this particular case, in the subspecies, we'll have in the species, we'll have wolf and a dog belong to the same species, and this is Canis lupus. The first letter of the genus will be always capitalized. Second one, please don't capitalize. So genus means Canis, where you will have a jackals, coyotes, dogs, foxes will belong to that as well. Canis lupus, Capus lupus. So now family, order, class, phylum, kingdom, and domain. You need to know those ones. And you probably remember from the biology classes in a school, in middle school, high school, or junior high school. King Philip came over for great, and whatever S is. The spaghetti, shoes, soup, or anything like that. In my time when I was teaching pre-AP and AP bio in one of the high schools, I found out that for 9 and 10 graders, sex works just beautiful. They remember that. They're like, King Philip came over for great sex. Like, sex, did he say sex? 9 graders. Yeah, I'm like, yeah, well, so in this particular case, make sure that you understand domain is the largest part. Today, actually, we'll talk about the domain prokaryote, and then the kingdom and so on. We'll talk about the kingdom Animalia later on. So from the smallest to the most inclusive, or to the largest, from the most inclusive to the least inclusive. All right, so make sure that you understand that one. Domain Eukarya, all of those creatures are in domain Eukarya, but now in Animalia, plants are not. Then we're talking about chordata, the one with the spinal cord, essentially, or cord, not a cord. So all of those except the insects. Insects considered to be invertebrate. Now we're talking about mammalia. Well, the fish is not mammal, so all of those creatures are more mammal. Well, all of them are mammals, and so on and so forth. All right, so let's kind of move on to the next one. So hierarchical species, genus, family, order, or in reverse. So domain is the most inclusive, while the species is most specific. As a matter of fact, in the real world, we have the only actual unit we have is species. Everything else is human-made. We are made it up. The genuses, the families, the names, and phylums, and kingdoms, and domains. Why are we doing it? This is one of the characteristics of human mind. We need to put everything in the different categories. So all of that particular thing are actually man-made. They are artificially human-created. Now, 3 domains. There is only 3 domains. There is tons of, well, there is like at least 5 kingdoms, at least probably 6, 7, up to 20 different kingdoms. There is over 100 different phylum. There is thousands of classes, and so on and so forth. There is 1 million of species. So now, 3 domains of life you need to know. Domain bacteria and archaea. We'll talk about that today very briefly during the lecture and then the lab, and we'll move on. If you're interested in that particular domains, you probably would be interested in a great class we have on South Campus known as microbiology. Really good team of 3 ladies doing an excellent job teaching microbiology, microbiology 1 and microbiology 2. Then eukarya. We'll spend most of our time talking about eukarya. By the way, for the microbiology, me in undergrad, it was 2nd, 3rd year course. Microbiology took me a year, 2 semesters to complete. I have only basically one day to go over the bacteria, so we'll go really, really briefly. We're not going too deep into that. And then, of course, eukarya. Eukarya will be broken down into 4 major kingdoms, even though most of the scientists know that probably protists need to be split up, and we'll talk a little bit about that next time. So in this particular case, prokaryote. Prokaryote means pro before, karyos means nucleus, before nucleus, no nucleus. Very simple, or the simplest type of the cells. Yet they have a cell membrane. Besides the nucleus, they don't have any membrane-bound organelles. If you look at the bacteria or archaebacteria, you will not find mitochondria, you will not find chloroplast, there is no endoplasmic reticulum or anything like that. So be very, very careful on what considered to be a prokaryote. It's not only the absence of the nucleus, it's the absence of membrane-bound organelle, and nucleus isn't one of those. All right. Now, eukarya, unicellular, and all of those are unicellars, are made out of single cells. Sometimes they're creating the colonies. That's pretty much no multicellular prokaryotes. Eukarya is single cell, colonial, and then multicellular. You are multicellular eukaryote. You belong to that particular group. Complex cells have a nucleus, have mitochondria. Some of them might have a chloroplast. We don't, but plants do. The endoplasmic reticulum, vacuoles, Golgi apparatus, etc., etc. All right. So here we have 3 major domains, and you need to know about those. Now, really quickly, I would like for you to answer really quickly a question. One of the most, or the most common cell in the human body is something which is known as a red blood cell. The one which carries oxygen, carbon dioxide, right? The one which makes our blood red, okay? So far so good, everybody understood that? This is the most common cell in the human body, red blood cell, tiny little bitty, very important to carry oxygen, everything is good. Okay, as the adult red blood cell, if you look under the microscope, you'll take a sample of blood, looking under the microscope, you stain it, everything is fine, you can see the red blood cell, you can see some of the membrane-bound organelles such as mitochondria and endoplasmic reticulum, but there is no nucleus. That's a true fact. You can look it up. Human red blood cells, or actually mammal red blood cells, don't have a nucleus. No nucleus in the red blood cell. All right? Just believe me on that one. Now, which particular group it will belong to? Is it a prokaryote or eukaryote? Answer that particular question really quickly. Prokaryote or eukaryote? Red blood cell. No nucleus, all right? I can guarantee you there is no nucleus in the human body red blood cells. If you are a human being, in the red blood cells, there is no nuclei. So, pro-eukaryote, type it in something on the chat. Activity, remember? Prokaryote because it's before. Well, that's the interesting statement. Anybody else? Prokaryote, okay, all right, cool, very nice. Anybody? So we have only 2 students who actually, 3 students who are, 4 students who are active out of 10. So everybody else, I can count absent right about now, right? Because you're not active. You're present, but you're not present. Need answers. I have 4 students only. Okay, 5. Prokaryote. Seems like majority of you think that the red blood cells are prokaryote because they don't have a nucleus. All right, let me rewind really quickly. Now, eukaryote, besides the nucleus, they're supposed to have a membrane bound organelles. Membrane-bound organelles, nucleus is one of those particular members of mitochondria, lysosomes, endoplasmic reticulum, Golgi apparatus, and so on. So membrane-bound organelles. Red blood cells, do they have a nucleus? No. Do they have a membrane-bound organelles? Yes. Meaning what? They are eukaryotic cells. That's one of your cells. If it's your cell and you belong to eukarya, all of your cells are eukaryotes. So make sure you understand that just the nucleus, present or absent, is not only the indication of pro or eukaryote. You're looking for the other things. It's one of the exceptions. And remember, biology is the science of exception. As a matter of fact, the cell which will develop into the red blood cell actually has a nucleus, but whenever it's mature, it's losing the nucleus because red blood cell lives for about 30 to 45 days, then it will be destroyed. As a matter of fact, your organism destroys about 1 to 2 million of red blood cells every single second. About 1 to 2 million of newly formed red blood cells from your bone marrow, and we'll talk about that during the unit 4, comes into your bloodstream to supply your organs with the oxygen. So keep that in mind. Not only the presence or absence of nucleus, but other organelles. Do they have a mitochondria? They have mitochondria, done. That's the eukaryotic cell. So here what we have, that's basically a tree of life. We already looked at the tree of life, started from some sort of prokaryotic organism, and then something happened and they start splitting up. And now it's split into the group of the bacteria, modern bacteria, and then there is an archaebacteria, ancient bacteria. We thought initially they were the first ones. Actually, no, a little bit they differently evolved than the eubacteria. And as a matter of fact, they're sharing some of our ancestry. Actually, we're sharing some of the genes that are closely related to us, to the eukaryotic organism, us right here, than the actual bacteria. And archaea is a relatively small group, and they prefer rather strange environment. For example, halophiles will live in extremely salty environments, methanogens, methanobacterium. Those ones live in the areas where there is no oxygen whatsoever. Thermoproteus lives in extremely hot environments, and so on and so forth. So in this particular case, as you can see, we're sharing the branch, or we shared the ancestor with archaea, and then archaea went and started developing, evolving, if you wish, in their own way, and then we ended up with the first eukaryotic cell, and that particular split happens, well, actually 5 billion of years old, almost immediately after the Earth is cooled down. So we're talking about 4.0, 4.2 billions of years, and we start seeing the liquid water, the life started on Earth. We don't know how exactly, and I'll talk about that a little bit later. But this is a very important split, and you can see that we had the common ancestor with archaebacteria later on in life. So, and this is where the eukaryotic organism, we believe, shows up on Earth somewhere around 1 billion or so years ago. So anyway, binomial nomenclature. Again, remember, unique scientific name, any species. Genus name is always capitalized. We'll have a, I probably need to make a capital. Species descriptor is not a capitalized. Both of the names italicized. You're looking at that particular tiny little bitty bug right here. It's not, it's a wasp. It's a tiny, about half-inch wasp. It will not be able to sting you. Females possess the stinger. Technically, it's the ovipositor, the one with the things which they laying eggs with. But that one, the ovipositor, is relatively soft. It will not be able to penetrate your body. So that particular, this is my favorite species out of all of them. You can say, why in the world that particular species is your favorite one? Well, here's the name of that species. Lindenius, that's the genus name. It's a group of the wasp, and that's Yakushkini. It's named after my last name. A friend of mine discovered that particular species, and as the present, he called me. He's like, yeah, we discovered the new species. I'm like, okay. And he's like, remember we went there and you collected those particular samples? I'm like, well, maybe, probably. And he's like, if you don't mind, I will name it after you. I'm like, sure, thank you. So that was one of my birthday presents, as a matter of fact. Whenever he published it, it was right coincide with my birthday. So yeah, yes, that's my favorite species out of them all. So average new species. We are continuing to discover new species, and then you can see in the last decade, well, basically from 2000 to 2009, over 17,000 species has been discovered. And remember, per year. And remember, we are killing about 1 species an hour without even discovering them. And of course, most of them, of course, are insects. And there is quite a few fungi. It's talking about fungi or hidden kingdom. We will talk a little bit more about that. And then, of course, the bacteria, but the classification of the bacteria is really, really hard. There is some aspect, but majority, the orange right here, are the insects. So phylogenic trees, we'll talk about that later on when we'll talk about Thursday in the lab. I'll talk about something which is known as the cladogram. But essentially, we talk a little bit about the cladogram before. Essentially, vertebral column, yes, no, breaking apart, creating the tree. Lancelet, one of the creatures, does not have a vertebral column, so no. Everybody else from that particular point, from a splitting point, will have some sort of vertebral column. Hinge jaw, lamprey will not. Everybody else will have. It's a special jawless fish. Legs. Fish, of course, don't have legs. Frog, lizard, rabbit will do. So you're always branching out. And this is what the cladogram is. And we'll talk about the, I will talk about the cladogram, and you will have a you will have an activity to do. So always branching out. Now, the one which is right next to each other, known as the sister taxa. If there is, if we have several species or groups, like several genuses splitting out from one common ancestor, that's known as a phylogeny. Basal taxon, just a single one. So it's one of the ways of classification and how we're creating with those particular species trees or tree of life, so to speak. Now, very important to see, we have a speciation event of some sort coming in, and this is where they split. So here we're looking at 3 different species, A, B, C. We had the common ancestor, which split into a species A, and species which later on will give us B and C. That particular species, let's call it species X, just for the for time being. So we have a species A and species X. Species X at some point of time had this split into a B and C. All of them are related. They all started with a common ancestor. However, species B and C closer related to each other than they are related to species A. All right, so we have a unique or common ancestor for the A, B, and C, common ancestor for the B as well as the C, and the common ancestor of C. So this is overall how we're looking at the trees, specifically when we're trying to show the ancestry. Now, few major things. A clade. A clade is a grouping which include all of the common ancestor and the descendants. So in this particular case, when you're looking just a single branch, it would be a branch clade. Now here, we're looking at the several branches, but because we're including pretty much all of the ancestors, now that's a clade. In this particular case, that's not a clade. And some students saying, why not? Well, because we didn't look, that particular species and 2 of those species had a common ancestor right here. If we would get all of that in the box, that would be a clade. In this case, it's not a clade because it's not including the common ancestor of 3 of those species. Here, it's not a clade because it's not including all of the descendants. It does not include that particular species. So here we have a clade, another clade, and another clade. Make sense? Kinda. We'll talk a little bit more on Thursday. So here we go. The birds and non-avian dinosaurs. What does it mean? It's basically the one which not turn into the birds. This is where your T-Rex will go. This is where the velociraptors are. But you see, they had the same common ancestor. Birds are showed up on this particular planet, coming from the dinosaurs, of course, but they went into their own branch somewhere around 225 millions of years ago. So in this particular case, again, talking about birds and reptiles. Now, reptiles, this is all of that considered to be reptiles. That's considered to be a reptile. Dinosaurs considered to be reptiles. Birds are not. So you see the classification of modern classification of reptiles should be split based on the cladistics, on the phylogenic classification system. But nobody really brave enough to break it down exactly. Well, because we don't know exactly what happened yet. We're still working on that. So here we have Dinosauria. You see, all of the clades and the birds are part of that particular Dinosauria. Now, whenever we're looking at the tree or cladogram of the vertebrates, you can see that some of the creatures are ceased to exist. Remember, 65 millions of years ago, dinosaurs, non-avian dinosaurs, T-Rexes, well, descendants of the T-Rexes, etc., died off for one or another reason. What reason? Most likely some sort of meteorite, supervolcano, both of them pretty much at the same time, killed not only the dinosaurs, lots of different species died off during that particular time. It was one of the mass extinctions. However, which give us the upper hand to mammals. And that's why 65 millions of years ago, after most of the dinosaurs ceased to exist, we start seeing the diversification of the mammals relatively fast taking place. So yeah. Shared character among all of the clade, and derived character, the one which will separate one clade from the rest. For example, 4 limbs, right? So sharks, in this particular case, we have a vertebral column. All of those creatures have a vertebral column. That would be a shared character. Now, 4 limbs. Sharks don't have 4 limbs. Therefore, it will be derived character for those particular creatures. Make sense? All right. And again, we'll talk about it. Homologous and analogous. We talk about that a little bit, but homologous versus analogous. Homo means same origin, might do a different functions. Look at this. Human hand, dog paw, the bird wing, and the whale. We had the common ancestor. Common ancestor started some sort of amphibians essentially, or maybe even fish, but a specific type of the fish. So in this particular case, our ancestors started it and we keep having it. Those particular structures are being modified. However, that we still can look at the same types of bones in all of those 4 rather different groups of animals. In this particular case, we're looking at the mammals. Well, that mammal and that mammal, the bird is actually the different class. So in this particular case, the characters different, but they have a similar similarity in the structure, meaning they had at some point of time, they had a common ancestor. Why all of us have 5 fingers or 5 toes? Oh, most of us, most of the mammals. Because we had a common ancestor at some point of time. We keep having it, even though, once again, right now you can do pretty much with only 3, and those particular fingers are reducing in size. The ring finger lost its ability to contract independently from the middle finger, and so on and so forth. They're sharing the muscle between 2 of those. Very interesting experiment. We might do it in the lab if you remind me. But in this particular case now, analogous, they start looking the same because they start doing the same function, but the wing of the bat and the wing of the bird evolved independently from each other. They look alike because they're doing the same function, same thing as, well, it's called the wing of the bee. In this particular case, it's something which helps them to fly. So analogous, similar looking, but very different evolutionary origins. Make sense? All right, so here we have, everybody knows a panda bear, right? Bamboo bear, and that's the primary source of that particular creature. Technically, it's not a bear, it's more closely related to the raccoons. But anyway, in this particular case, human has a thumb. What does the thumb does? Helps us to grab something. So the panda bear, whenever it grabs the bamboo and putting it in the mouth, had developed as something which is known as a full homologous structure. Answered in your chat box. Let's see, what do you think? Is it analogous or homologous to the human thumb? You can put A or H if you don't want to type it in, analogous or homologous. So let's see. A or H. So thumb to human thumb. Is it the analogous structure or homologous structure? Homologous. So they have same origin. Analogous. So they do the same function or same origin. Very simple, by looking at the, let's kind of look at the picture before I'll give you the correct answer. Let's count how many toes or fingers the panda has. 1, 2, 3, 4, 5, including the 6 of the false thumb. So if it would be, and how many humans has 1, 2, 3, 4, 5? It would be, if it would be a homologous, same origin, that particular thumb have to be number 5, right? Yeah. So it would be, as majority of you says, analogous structure to do the same function, to grab something. an opposite thumb to grab it easier. So in this particular case, false thumb would be the analogous. That's why it's called false. So be careful with that. Now we're looking at the two different types of the evolution, convergent and divergent evolution. Convergent evolution, it's not when the two species merging together, even though name kind of predicting. It starts saying that the species start looking the same or looking very similar to each other because they're doing the same function in the nature. Think about shark and the dolphin, right? So they look very similar to each other, streamline body, the fin on the top, and so on and so forth. Why? Because, well, both of them are top predators in the same environment, in the ocean. They have to have a fast speed in order to grab whatever the food they have. If we're looking at the if you'll ever be able to see them in the wild, the dolphins versus sharks, first you see, like I'm not really sure if it's a dolphin or a shark, because you'll see the relatively dark bodies lurking in the water, and then there is the that particular top fin. Both of them has it. The difference is if you'll see two top fins, the top fin and then the vertical fin or tail fin, that's the shark. The purpose or the dolphins, they have a horizontal tail fin. Anyway, it creates the problem. For example, let's take a look. Well, homologous versus the analogous structures. The arm of the octopus or the arm of the sea star or the leg of the grasshopper. Do they have exactly, if you will look at the, if we'll cut it up, the grasshopper leg, does it have exactly the same structures or features as we have? No, it does not even have a bones. They have an exoskeleton. The skeleton is outside of the body. Sea star has an endoskeleton, but that will not have those particular bones as we have it in our legs. So it will be analogous structures. They come up independently from each other. They develop independently from each other. And this is where we start talking about the origin of the head. What head is? Head is the front part of the body which goes into the environment. As a result, we start seeing the concentration of the sensory equipment, so to speak, sensory, any kind of sensors on the front end of the body. Sensors receiving any kind of signals from the environment, the light, the sound, the chemical ones, etc. And then we have to have a processing unit, the brain. So the head of the flatworm and the head of the vertebrate probably evolved independently from each other. Same thing as the head of the octopus, well, the head of the mollusk or the head of the arthropods. Same idea. Front end goes into environment, have to sense it. As you are sensing, receiving the information, that information needs to be processed. So analogous structures, even though they call a head because it's a front end, it has the most of the sensory equipment, the eyes, the ears, and so on. Well, not necessarily ears. For example, cockroaches, insects. Many of the insects have the ears on the legs, so to speak. So anyway, not homologous. Homologous. Why? They're doing the same function, but the structure is very different, right? And then we have, oh, here we go, homologous or analogous. If you will be ever in the South Campus or in the science building, you will have this particular, well, we have the, on the first floor, we have the skeleton, a full skeleton, or replica, of course, of the Similodon, the tiger, well, the saber-toothed tiger, so to speak. And then we discovered another creature, which was Stylacus milus, and initially they were put together as the group of the saber-toothed tigers. When we start looking a little bit more closely, we ended up that these creatures, well, they distantly relative to each other, but those saber-tooth developed independently from each other. They didn't have a common, well, recent common ancestor, because one of them is actually a marsupial, another is the fully placental mammal. Once again, the appearance is a convergent evolution because they were top predators, killing something which is known as a megafauna, right? A large kangaroos, like for example, that one was killing a large kangaroos, and those particular kangaroos were like 10, 12 feet tall. In order to kill that particular beast of a kangaroo, you have to have a very sharp and large canine, so to speak, even though it's a cat, right? And same thing with the similodons or saber-tooth tigers. They evolved in order to kill a larger prey. So they had a common ancestor, but those teeth developed independently from each other. Well, the large teeth, or the origin of the saber-tooth, as you can see, it would be a homologous. They start converging. Now, diverging, we already talked about that a little bit more. Common ancestor and then split. Some of the modern elephant stays either in Asia, or tropical Africa, evolved in the modern. Some of the groups of the common ancestor ended up somewhere up north in Asia and North America, and they evolved into a woolly mammoth, which is a completely extinct, predominantly, well, climate change and humans overhunting those particular creatures. We're doing the same thing with overhunting for the modern elephants. The African elephant is the protective species, and if the poaching continues within the next 20 years, there will be no African elephants in the nature. Still in the zoos, but not in the nature. So here we talk about we talk a little bit about the evolution of the hominids, common ancestor, and then we start splitting up. And as you can see, in this particular case, old world monkeys, we didn't evolve from the monkey. That's one of the very important things you need to understand. Humans did not evolve from the monkeys. We split from the common ancestor. We had the common ancestor of those particular, with those particular common ancestor of modern monkeys about 35 millions of years ago. And then there was a branched out different species. Some of them died off, but eventually around 10 or so millions of years, we had a common ancestor with modern gibbons. And again, it wasn't like branched out and 9 millions of years ago, we have a modern gibbons. No. The gibbons went their own evolutionary history, so to speak, to turn into a modern gibbon. Same thing with orangutans, and as you can see, the human and chimpanzees are actually gorillas split a little bit earlier than humans and chimpanzees. We close related to humans and chimpanzees are the most closer related species, but we did not evolve from those creatures. Please do not give me that particular example. Please, it drives me insane. I'll start typing in the capital red letters, we did not happen, etc., etc. I have students who still believe that humans and chimpanzees, well, chimpanzees evolved in humans. No, that's never happened. It didn't happen at all. Please, if you learn one thing in this particular class, that we did not evolve from chimpanzees, please don't give me that example, ever, or anybody else. So human evolution, we already talked about that in the lab. We talked about the actually several major groups. The orange one represents, of course, us, Australopithecus, some type of Australopithecus. Then we head into a Paranthropus. Remember the guys which went all muscles, and here we had all brains. There was quite a confusion on the filling out the chart on the human evolution, or specifically for the Paranthropuses. Most of you saying they have the sagittal crest, the brow ridge, very massive bones, meaning and very large the zygomatic arch, so very large jaw. But some of you says that they have a really small teeth. That's not true. If you think about it, if you have a sagittal crest, a brow ridge, it means that you have a huge chewing muscles. If it has a huge chewing muscles, the teeth have to be large to withstand that particular pressure. Because if for one or another reason, think about a modern gorilla, if we'll take modern gorilla, huge chewing muscles, and we'll replace gorilla teeth with the human teeth, same skull, first chomp, the teeth will literally crack down. Why? Because there is so much force, they will not be able to withstand. In order to withstand the force, the teeth have to be relatively large. Besides, the small teeth will worn out much faster. I'm grossly exaggerating, but still, this is the general idea. So Paranthropus was a dead end, and look right here, some of them survived up to like 800,000 years ago and then died off. Or did they die off? Some of the scientists saying that some of them are still alive, and this is what the Yeti is, the Bigfoot thingy. Maybe, I don't know. Maybe they're still alive. Maybe they're just a very scarce population. Now we're looking at of course, at the Homo ergaster, Homo erectus, maybe all together, not 100% sure, but now we're looking at the Homo erectus, could be the same species, but look, they survived for over 1 million of years, for over 1 million of years, were a very successful species, and when it died off, when the humans showed up. Essentially, one of the theories, and it's a theory, saying that whenever the humans originated in Africa, we start actively hunting those particular guys because you're coming in, you destroy, well, you kill those creatures, probably eat them too. But also they were making the tools so you can take over the campsite of those particular creatures. And then this is the creature which actually moved out from Africa into Asia and Europe. So, and this is one of the reasons why humans decided to move out. And of course, whenever we moved out from Africa, we ended up with Neanderthals or meeting the Neanderthals, and eventually we killed them. So yeah, that's for sure. Anyway, co-evolution, another very important part of the evolution, when two or more species start evolving together, one changing and another changing in response. Anybody knows what you're looking at right here on the left bottom corner of that particular thing? That's two different species. They're close related to each other, but what kind of species are that? Anybody ever seen those creatures? You probably heard about those. I'm certain you heard about those. That's lice. Yeah, almost. Yes, you are right. It's an insect. It's called lice. Yes, there is two. It's actually human lice. Yay. Yeah, I know. Yeah, don't have lice, as far as I know. Anyway, in this particular, we have a head lice, and we have something which is known as a pubic lice. What lice does is holding on, it has that particular claws to hold on to the human hair. At some point of time, whenever our ancestors were completely covered, it's not humans yet, completely covered with the fur, it was a single species. Then we start losing in order to better cool down, etc., we start losing most of our hairs, and we ended up with two major areas still covered in the hair. It's a pubic area, and you might know that the pubic hair is usually thicker. Look at those particular pincers. Those pincers are evolved to hold on to a very thick hair. And we have a head hair, which is much thinner. Therefore, the pincers are smaller. If you'll take the pubic lice and put it on the head, it will try to grab onto the hair, but it will literally slide off. So now evolution within the human ancestors, it wasn't humans yet, evolution within the human ancestor lead to the evolution of two now different species of lice, which have a geographic barrier. Literally, some of them are living in the area of the head, and others down there in the pubic area. They do not cross much between each other. Now, and we ended up with something which known as the merging of two species. They close related, the superlice, which can live pretty much anywhere. So that's one of the examples of the coevolution. Our hair changes and the lice changing as well. Now we're looking at this particular bird known as the crossbills. Crossbills, it has a bill which is kind of crossed in order to open up those pine cones relatively easily in order to get the seeds. And of course, the pine does not really like when the bird is eating the seeds, so it cannot spread. So the pine cone changed. It become bigger. As it's become bigger, so those particular species of the crossbills cannot reach into those particular cones. So they had to modify, evolve, or co-evolve with the modification. Now they come up with a bigger, stronger bill. And then, of course, there is something which is known as a mutualistic species. Mutualistic species, this is two species which are helping each other. Before we were looking at predator and prey, or parasite and host. Predator, the antelope become faster, the predator will have to become faster as well. Now, on the mutualism, oops, hold on guys, let me kind of pull it off really quickly. On a mutualistic relationship, let's say the insects, butterflies, and the plants. Butterfly comes and the plant has the flower inviting the butterfly, like, hey, come, there is food for you, the nectar. The butterfly comes, everything is fine, getting the nectar, feeding itself, or the bee or the bumblebee, whatever. Doesn't really matter. But what the plant does, so the insect is benefiting getting the food. What the plant does, plant sending or showering that particular insect with the pollen, which is essentially a plant sperm. We'll talk about that during the unit 2. And next time when the bee or butterfly goes to the next flower, it transferring the sperm from one flower to another. Cross-pollination. So both of them benefiting as a result. The plants evolved along with the insects, inviting them, giving them something sweet to eat, and so on and so forth. So very important. So coevolution, two or more species working together, all right, affects each other evolution. All right, well, let's kind of leave it as is, and let's go into a new lecture. Should be available to you already, and that's talking about the I need to swap. Here we go. So prokaryotes, a living organism. And here we're looking at some of the things. This is a hot spring from Yellowstone. If you'll ever be, I will in that particular area. I would highly recommend you to go to Yellowstone. Really, really interesting. Almost some of the areas looks like you are on a different planet. And this is a hot spring at about 80-90 degrees Celsius. Water meaning almost boiling, about 200 degrees Fahrenheit. And that particular brown stuff is bacteria living there, specifically archaebacteria. And then, of course, all of you heard about genetically modified organisms, right? GMO. That's bad. We don't really know. We are pretty much lab rats to the big companies who are introducing genetically modified organisms as a part of our food sources, corn, soy, tomatoes, etc. And we don't know what's the long effect of the genetically modified organism. Some people saying it's bad, some people saying there is no problem. So we'll talk about that a little bit more. So here we have a genetically modified mash. And then all of you heard about E. coli. the bad one? Well, technically all of us have an E. coli in our digestive system right now, but some of them might get genetically modified due to mutations and turn into a bad, which will affect some of the, well, which will start getting, instead of being just in the digestive system, start moving into the kidneys and killing the person. So fossil record, extremely important. And today in the lab, we'll look at those particular things known as the stromatolites. What the stromatolites are? This is actually the living stromatolites of coast of Australia. As you can see, shallow ocean. And this is the rocks right here. This is a bunch of rocks. At the very top couple of layers are living cyanobacteria. Cyanobacteria are 3.5 to 4 billions of year old bacteria. Well, we know that we have some fossils of the cyanobacteria, which is about 4 or so billions of years old. But what happens? The bacteria, that's a photosynthetic bacteria, and today we'll talk about the photosynthesis, one of my favorite reactions. I'll explain it momentarily why I said favorite reaction. So anyway, what happens? We're looking at the strands, the colonies. Each one of those particular cells are separate bacteria, but they stay together, creating a filament. So eventually we'll end up with the map of the bacteria. So far, so good. That bacteria getting the sun, photosynthesizing, getting food, everything is fine, all good. Whenever it has plenty of food, it starts dividing. As it divides, some of the layers will cover the previous ones. So we have a layer of another one on top of it. They divide it, right? So far, so good. So it will create a layer, and then another layer, and then another layer. So the bottom layer now stop getting the sunlight. It cannot photosynthesize, it dies. Whenever it dies, it's creating that particular mesh where some of the sediments from the shallow ocean will get trapped in, and it will create those layers in the stromatolites. And today in the lab, you will see those particular layers. Stromos means layered, and the lithos means stone or rock, layered rock. So it will create, and as basically as it happens, of course, it takes a long time, but they start growing. They kind of getting up to the level of the water. So in this particular case, we're looking at the stromatolites, living stones, because on top of them, they covered with the still living bacteria. The bottom one will be a dead bacteria, which created a mesh, and all kinds of sediment get trapped in that. That's one of the reasons why the layers will be slightly different colors. So those are extremely important bacteria. Why? Because they started with the process of photosynthesis. Photosynthesis, those cyanobacteria about 4 billions of years ago started the process of photosynthesis, releasing oxygen in the atmosphere. 4 billions of years later, we're looking at the oxygenated atmosphere. We have a lot of oxygen in the atmosphere due to, well, they started it. So very important. The oxygen which you're breathing right now probably come out from the bacteria 4 billions of years ago. So now, size of the prokaryotic cells. All of you, if you took Bio 1, you did that particular experiment when you are taking the cheek cell, putting it on the glass slide, you're staying with methylene blue, covering and looking under the microscope. And in this particular cheek cell membrane, you can clearly see the nucleus, you can see the cytoplasm. Everything is fine. However, and that's one of the actual slides from the Bio 1. Some of the students start asking, what about those tiny little bitty dots? I'm like, well, that's the clusters of bacteria. Like, eee. Bacteria. I'm like, yes, you have a bacteria. You need 3 things to grow. It needs a relatively warm and wet environment and plenty of food. Our mouth is a perfect breeding ground. Warm, wet, and anytime you are consuming it, particles will stay in your mouth. So here we go. Bacteria can reproduce every 20 minutes or so, giving the good environmental conditions, meaning that we literally have millions of bacteria in our mouth right now, even though, yes, that's another ew. Even though I brush my teeth today, I brushed my teeth last night, and so on and so forth, I even rinse it, yet still millions of bacteria. For you guys, I hope we don't have much of the germophobes. Within the regular kiss, you're receiving about 5 to 10 millions of bacteria from your partner. You're welcome for that particular piece of information. Well, there is this there is a silver lining to that. About 5 to 10 millions of bacteria on average leaving your mouth and goes to your partner. So it's like mass migration of the bacteria. Let's kiss. Well, let's mass migrate some of the bacteria. Yes. So here we go. Within a handshake, a typical nowadays it's almost prohibited, right? You're doing all kinds of weird things. But within a handshake, somewhere around 15 to 20 millions of bacteria goes from your hand to another hand. Yeah. Oh, vice versa. So you're receiving. So anyway, as you can see, most of the bacteria cells are tiny, very, very small, about 1,000 or so times smaller than the typical eukaryotic cell. So here we have a eukaryotic cell. Here we have a prokaryotic cell. Very, very simple, tiny, little bitty ones. Three technically four major types of the morphology. Bacteria, we figured it out that some of them and the microscopes were not as good as we have it right now. But basically, the scientists subdivided the bacteria into four basic forms, 3 or 4. The first one is the round one, that one known as a cocci. Single one will be coccus, and the plural will be a cocci. Then there is a rod-shaped bacteria. Today in the lab, you will see some of those examples. Those ones known as a bacillus or bacilli in plural. Okay. Then there is a spiral one. It's known as either spirili or spirochete. I know spirochete, the misspelling can be happening on the lab practical, so make sure that you know either spirili or spirochete. Speaking of which, some of the students are very concerned about, do I deduct for misspelling? Not the greatest speller myself, so it would be kind of hypocritical for me to do the misspelling errors. If I can read and understand what it means, spirochete, let's say you left the H and A, spirochetes or C, whatever it might be. If I can understand that you're talking about the spirochetes, I will be okay. I'm not a grammar Nazi, so to speak. So once again, and it's not only English, because English is my second language. I'm not the greatest speller in the Russian either. The only B I had in the high school was Russian language. The only B in the university undergrad I had was a Kazakh language. So anyway, so languages is not my forte to begin with. So in this particular case, as long as I understand what you're talking about, however, be very careful. In some of the terms, it could be very close. One of the examples in Bio 1, I do remember example, and I told the student, I put the smiley face. She misspelled Golgi apparatus. There was a first lab practical where you're supposed to name the structures of the cell, and the arrow was on the Golgi apparatus. And I was looking Golgi apparatus, Golgi body, any of those particular things, right? Golgi, that was the keyword. She misspelled it into a gigolo apparatus. And that became a very, very totally different thing. It's not even a cell-based. So be careful with that, all right? So, but yeah, if I can read it, Australopithecus or Paranthropus, That wasn't, could be, especially if it's an afarensis. Australopithecus afarensis kind of have a tendency to be misspelled, but in the lab practical, if I can understand that, I will give you, I'll go back and I'll assign, speaking of that, every single lab practical, I will go and will reassign any of those points which needed. All right, we got the spirochetes. Now, the last but not least, known as the vibrios. Vibrio is like a spirochete. It's starting like a spiral corkscrew, but it's just a tiny little bit. It's like comma shape, or think about it as a small sausage, curved like a comma. It's starting into the wavy corkscrew, but it just stops. Usually a small one. Today you will see the color. I think color is the vibrio. So this is 3 or 4 major forms: coccus, bacillus, spirochete, spirillum or spirili, and the vibrio. All right. Now, the structure of the or features of the cell. Any kind of cell, no matter eukaryotic or prokaryotic, will have several main parts. One of the parts is something which is known as the cell membrane. Double layered, it's all identical, pretty much same as the for eukaryotic or prokaryotic cell. This is the separation of the inner part of the cell with the outer environment. So all of the cells, plant, animal, fungi, prokaryotes, will have a cell membrane. The structure is almost identical among of those particular groups. So cell membrane, make sure you understand that. Now, inside it will have a cytoplasm, kind of jelly-like material where everything is dissolved. Now, the smallest and most numerous organelles in any kind of cell again. So all of the cells will have a cell membrane, all of the cells will have a cytoplasm, all of the cells will have what? The organelle which produces protein or makes protein. Anybody? Anybody? No? No? Don't remember? Which organelle produces proteins? Helps making proteins. Actually, the maker of the proteins, known as the ribosome. So all of the cells, prokaryotic and eukaryotic, will have those 3 things. What's really interesting, I don't know if your Bio 1 teacher told you about that, the kind of controversy, egg and the chicken. The ribosomes are responsible, this is what they sole responsibility, to make proteins, taking amino acids, grab the messenger RNA and putting it together. So far, so good. So they are responsible to making proteins. They are the makers of the proteins. The ribosomes are made out of, well, guess what? Proteins. So in order to make a ribosome, you have to have a, well, in order to make a ribosome, you have to have a protein. In order to have a protein, you have to have a ribosome. Which one came first, protein or ribosome? Anyway, moving on. And then there is a DNA strand. It's usually a circular DNA. As you can see, it lays down straight in the cytoplasm. It does not have a membrane around it. No nucleus, it mentioned. No, you don't. Do you see a mitochondria? No, you don't. Lysosomes? No, nothing like that. Very, very simple structure of the cell. So we have a ribosomes, we have the cytoplasm, we have a DNA, sometimes referred to as a chromosomal DNA. It's basically the circular DNA in the cytoplasm. In addition to that, sometimes there will be a tiny little bitty circles of DNA in addition to the main one known as the plasmids. I think next picture is talking about that. Now, bacteria, in addition to that, will have a one or several flagella in order to move. Some of them don't, so they don't really move. Then they will have something which is known as the cell, right? That one is the cell wall, the kind of darkish green. So green is a cell membrane, but then cell wall. Cell wall will be made out of a very specific chemicals known as the peptidoglycans. We'll talk about that momentarily. And they're very important. Not all of the cells have a cell wall. And the cell wall quite often is very specific for a particular group of creatures like bacteria. The cell wall will be made out of peptidoglycans. The plants, the cell wall will be made out of cellulose. Fungi, the cell wall will be made out of chitin. Animals don't have cell wall. In addition to the cell wall, many of the bacteria will have something which is known as a capsule. That's additional layers of protein and lipids typically to increase the adhesivity to the surfaces, to stick to the surfaces. And then from that, you will see the hair-like projections coming from the cytoplasm, known as a pili. Single pilus, plural pili. That's basically to increase the surface area. Why do we need to increase, or why the bacteria need to increase the surface area? To increase absorption. They're feeding from, most of them, feeding from the whatever they can get from the environment. So those particular structures, and we'll talk about those structures in the lab as well. So here what we have, cell wall, cell membrane, orange, cell wall, purple, very important. And then we have ribosomes, and then we have the central DNA, and that's a plasmid, that additional DNA, which is not a part of the main DNA. Now, cell wall. The structure of the cell wall, I already told you about the peptidoglycan. Peptido means proteins, glycan means glucose, the short chains of the sugars intertwine with the layers of the protein, peptidoglycan. All of the bacteria will have a cell wall made out of peptidoglycan. Some of them will have something in addition to the peptidoglycan. And based on that, there is something which is known as a gram-positive and gram-negative bacteria. Gram was a gentleman microbiologist who came up with the idea how to stain the bacteria cells. Why do we need to stain the bacteria cells? Without the staining, the tiny little bitty one, the transparent. Without staining, you will not be able to see them. Once again, you did that particular experiment in Bio 1 when you are doing the cheek cells, and if you put without the adding the methylene blue, you don't really see anything. They are transparent, they small. So in order to see the cell or structures within the cell, you need somehow to stain them. And the Gram come up with the staining technique. And if you'll go into microbiology, you'll learn that particular technique. I don't have time to go over it. Just think about that the staining technique, there is a special stains, dyes, which go and attach themselves to the peptidoglycan to make it visible under the microscope, of course. So, and according to the Gram staining technique, the color of the cell wall have to be purple. So if you'll see a bacteria which stained by the Gram technique, kind of bluish purplish color, that's a Gram-positive bacteria. Done, everything is fine, no problem whatsoever. But Gram noticed that some of the bacteria will not stain fully into a purple color. Some of them will be non-purplish, generally kind of reddish pinkish color, sometimes brownish color, basically non-purple. So he called those particular groups of bacteria Gram-negative bacteria because they're not fully accepting the staining technique. At that time, and it happened about 150 years ago, he could not explain why. Now we do. The main difference is in the cell wall. All right, Gram-positive bacteria. So cytoplasm of the bacteria, the inner part, the double-layered plasma or cell membrane, and then the cell wall made out of what? Peptidoglycans. We have the chains of proteins interlinked with the sugar chains. Everything is fine. So whenever this stain comes from the Gram staining technique or stains, dyes, they will stain that particular part of the cell wall. So far, so good. Cell wall is made only out of peptidoglycan wall. Any questions, any problems on that? On a gram-positive bacteria. It will be on a lab practical, it most likely will be on a test. Now, on a gram-negative bacteria, what do we have? We have exactly the same thing, cytoplasm, cytoplasm. We have exactly the same plasma membrane. Everything is the same between the bacteria. Now, when we start looking at the cell wall, it has a slightly different structure. It's more complex. It's like, think about it, this is like a piece of bread. Here we have a piece of bread, but now in addition, we have other things. We have a sandwich in this case. We have a peptidoglycan, still a cell wall, but then we have something which is known as a lipopolyproteins or lipopolysaccharides. In this particular case, what does it mean? We have a cell wall which is made out of several layers. So whenever the stain tries to go through that, it will not be able to. So it's staining not very well the peptidoglycans. As a result of that, as a result of the structure, more complex structure of the cell wall, those particular bacteria will not be stained nicely purplish. They will be other than the purple color, usually brightish pinkish color. Make sense? So make sure you understand. If you'll see on the two pictures without any gram-positive, gram-negative bacteria, if you'll see it on today's lab, there will be a structure of the gram-positive and gram-negative cell wall. The difference is in the cell wall. Both of them have a peptidoglycan, but this negative, gram-negative bacteria have something else, lipopolysaccharides. It's basically like an additional cell membrane on top of it. The structure is very similar. I'm not saying it's a cell membrane. It's just like a cell membrane. As a result, if you think about it, let's say we're treating two of those bacteria, gram-positive and gram-negative, with antibiotics. Antibiotics slowly making its way through the peptidoglycans, through the cell membrane and the cytoplasm, killing the bacteria. It's toxin, essentially. Now, if we're doing this, first they have to penetrate that, then they have to penetrate that, then they have to penetrate that, and then they will have to kill the bacteria. Where the antibiotics will be working faster, in a gram-positive or gram-negative bacteria. Gram-positive because they have less stuff to go through. Just to go through the peptidoglycans, cell membrane, done, you are already in the cytoplasm. Here you have to go through the lipopolysaccharides, then the peptidoglycans, then the cell membrane, and then only then into a cytoplasm. So the structure of the cell wall is, this is what gives the different colors to gram-positive and gram-negative bacteria. Lots of students historically, I'm not saying you are, but historically are kind of confused on that. So here we have a cell wall is made out of just the peptidoglycan. And here we have a peptidoglycan, but something else in addition to. It's sandwiched, it's layered. There is another layer or two of lipopolysaccharides. All right? Now let's talk about sex. I mean reproduction. Yes, we always will be talking about the reproduction sex within the group of organisms. A very simple one, binary fission. The cell splits into two daughter cells. And see here, I can already say, well, that's the sexist, because daughter cells. There is no father cells or son cells, right? My cell, I'm a male, presumably, biologically at least. So in this particular case, whenever my cells are divided, they create a daughter cells, even though I'm male. I want to have the maybe son cells. Just kidding. Actually, if you go in the German I don't know, you might actually tell me in Spanish. Is this in Spanish will also, the result will be a daughter cells? In German, in French, in English, as well as in Russian, it would be daughter cells. So seeing clearly the males are being underrepresented right there. Anyway, again, that's talking about the social aspect. So binary fission, splitting into identical cells, very fast, known as the asexual reproduction. No sex per se. That's why I said we'll talk about sex. I mean the reproduction, because not all of the reproduction is sexual. So that one is asexual reproduction because the daughter cells are identical. They clones of itself or each other. So, oops, let me go back really quickly. That happens really, really fast. Binary fission is very similar to something which is known as a mitosis. Remember, in Bio 1, you probably talk about the mitosis. Why it's not called mitosis, why it's called binary fission. Because mitosis, and then later on cellular division, mitosis is the splitting of what? Of nucleus. They don't have nucleus. It cannot be called mitosis. No nucleus. That's why there is a binary fission. One big cell splits into two identically, genetically identical clones of each other. The bacteria can do really, really fast. Every 20 minutes a generation. 2, 4, 8, 16, 32, 64, 128, 256, and so on and so forth. Meaning what exactly? Remember our mouth, I'm going back to that particular thing. You brushed, you brushed, you brushed, you swish, you swish, you swish. Everything, you rinse it, everything, all of them died except one. One was somewhere hiding out. So now that particular one, in less than 24 hours, will end up with more than 1 million of bacterial cells. You can calculate it every 20 minutes and I'll give you the progression. Now, there is 3 other mechanisms which now considered to be a sexual reproduction. Why? Because in the sexual reproduction, the daughter cells or resulting cells will be genetically different from the parent. This is what essentially sex in the biology talking about, whenever the offspring is genetically different. So now let's take a look on those ones are rather weird part. Yes, I know we are kind of a little bit over the time, but bear with me, we're almost done. So transformation. This cell, bacterial cell, swimming happily, everything is fine, everything is good, life is good, it's dividing, binary fission, everything's good. When it will get into extreme condition, whenever it's stressed out, the bacteria has the option to absorb, literally absorb DNA from the environment and incorporate that particular DNA into their own DNA and start producing whatever that piece of DNA was telling them. Why? In a vain hope that that particular DNA will help them survive that particular stress. That's called transformation. This is how we're making genetically modified organisms. For example, you have a bacterial cells. You have the human DNA, which is coding for human insulin. You're stressing the bacteria out. How? You yell at it. No, no, no. All you need to do is take that particular mixture in the test tube. You're putting it in the warm water, not boiling, like, all right, let's say around, but not comfortable for the bacteria. Bacteria are pretty good at about 90 to 100 Fahrenheit. That's the threshold for many of the bacteria. You're putting it in 110 degrees water, the test tube, for a couple of minutes. And then you're putting it into ice water. So global warming, global cooling, global warming, global warming, global cooling again. The bacteria start getting stressed out. It starts absorbing the DNA, human insulin, DNA, the DNA which coding for human insulin. Now you have that particular bacteria which producing human insulin. Human insulin well, that particular protein cannot be used by the bacteria, so it secretes out. You are multiplying by binary fission that particular transformed bacteria, genetically modified with a human insulin gene in it. It's producing a lot of insulin because of the diabetes. All right. So this is why the bacteria is so adaptable. There's the next one, which is similar to a transformation, but in this particular case, it has vector, the thing which will bring the DNA inside of the bacteria cell. And this is the viruses. There is a viruses known as bacteriophages, which can kill the bacteria. However, sometimes whenever the virus is injecting its own DNA, it's a mutated DNA. The virus no longer functional. It will bring a new genes to the bacterial genome. As a result, the end result would be genetically different cell. So it's not died, the virus was mutated, it wasn't inactive, so to speak. So, but now the daughter cell is genetically different, sexual reproduction. So we have a one-way transformation, then we have transaction, and last one is conjugation. Now, this is the closest to bacterial sex we can have. Basically, the pili, they merge. Remember, pili extension of the cytoplasm to increase the surface area. So there is a special type of S or sex pili, they merge, and the bacteria will exchange some of the pieces of DNA with each other. Actually, from one bacteria will donate another. So we start talking about the donor of the DNA information, male to a recipient, a female. All right, and that's a conjugation. That's basically, the last one, endospore. Now, bacteria, again, warm, wet environment, plenty of food. If any of those particular things start deviating, not enough food, it starts getting too cold, drying out, some of the bacteria can form something which is known as the endospores. Essentially, it's a very complex process, but essentially the cytoplasm will kind of create the layers around the core, and in the core it will have a DNA. And as the spore, the bacteria can withstand the drying out, completely freezing out, and just wait for the really good environment to come back to life. So that's to protect genetic material during the extreme stress. So again, cold environment, dry environment, not enough food. And we'll talk about the endospores in the lab momentarily. All right, phylogeny we'll cover next time. And we're done with the lecture today. Let me stop share and let me stop recording.